Hello everyone, this is the third lecture of Module 2. The topic of this lecture is glandular trichomes and cannabinoids. Let me explain what a glandular trichome and cannabinoids are. Cannabinoids are and terpenes are synthesized by secretory cells and stored in the secretory cavity of a glandular trichome. A glandular trichome has a mushroom-shaped structure under magnification as shown in this slide. There are three types of cannabinoids. Endocannabinoids are endogenous and occur in our body. On the other hand, phytocannabinoids and synthetic cannabinoids are exogenous. Plant-derived cannabinoids are called phytocannabinoids, including CBD and THC. Synthetic cannabinoids were originally developed for research and do not occur in nature. In this lecture, I will just talk about phytocannabinoids. Phytocannabinoids are unique compounds found in cannabis plants. More than 130 phytocannabinoids were currently identified. The most common cannabinoids are delta-9 tetrahydrocannabinol, called delta-9-THC, and cannabidiol, called CBD. THC is responsible for marijuana's psychoactive effect, whereas CBD plays a more modulatory role and can't intoxicate. As a transition state molecule, dozens of minor cannabinoids, including CBG and CBC, they have unique therapeutic potentials. Let's look at the structure of glandular trichomes. A glandular trichome consists of a globose head, including secretory cells, stipe cells, and an enlarged secretory cavity. And there are many secretory vesicles in the secretory cavity. This slide shows us the biosynthesis of cannabinoids. Olivethoric acid is a major intermediate in the biosynthetic pathway of cannabinoids. At the first step, olivethoric acid and the terpene unit create a CBGA. Sequentially, CBGA converts into THCA or a CBDA. So CBGA is the foundation of all cannabinoids and is called mother cannabinoid. Let's look at the chemical structure of two major precursors. Olivethoric acid is a phenol compound, and it has a benzene ring with two hydroxyl groups, one carboxyl group, and a pentyl chain at the position of carbon 5. Another precursor, geranyl diphosphate, is consisted of monoterpene group formed from two isoprene units and a diphosphate group at one end. The combination of these compounds form a CBGA. CBGA is the first cannabinoid to appear in the cannabinoid biosynthesis. As I mentioned in the previous slide, CBGA is created by combining an olivethoric acid and geranyl diphosphate. There are three chemical forms of cannabinoids. cannabinoids are produced in the form of acid. These acidic cannabinoids are low and carboxylated forms. They are mainly non or less psychoactive. For example, THCA, CBDA, and CBCA. However, neutral cannabinoids are decarboxylated forms, which have lost carbon dioxide. They are biologically more active. For example, THC, CBD, and CBC. Finally, degraded cannabinoids are transformed by oxidation, heat, or light. They are less psychoactive. For example, CBN and delta 8 THC belong to degraded cannabinoids. Phytocannabinoids readily transform from one form to another during the biosynthesis in the plant and the storage period after harvest. There is an important concept in the cannabinoid industry called entourage effect. The entourage effect means that all the natural chemicals work better together than any single compound alone. For example, if you mix CBD and THC in a 2-to-1 ratio, the psychoactive effect of THC disappears. THCV can cancel out the appetite-stimulating effect of THC. Also, alpha-pinene preserves acetylcholine, which prevents the short-term memory loss caused by THC consumption. By the color of the glandular trichomes, it can be identified its maturity. Glandular trichomes can be classified according to their secretory phase from the color of their contents. Immature glandular trichomes are translucent in appearance, while aged glandular trichomes are the peak time of cannabinoid production, appearing cloudy or amber. At the stage of senescence, major cannabinoids start conversion to the chemically stable cannabinoids such as CBN. Actually, we investigated how the amount of cannabinoids in the glandular trichomes differ depending on the stage of maturity. When mature stage, CBDA content is peak and the trichomes appear clear. During plant aging, CBDA concentrations decrease. On the other way, THCA gradually increase. When senescent stage, THCA content is peak. However, the concentration of CBDA is at its lowest. Examined both sessile and stalked glandular trichome for their respective cannabinoid contents. Whole glandular trichomes were removed from bracts and the leaves of the same plant to compare their contents. They were extracted for cannabinoids and analyzed. Results show that the stalked glandular trichomes on the bract contained more THC, approximately 20 times more than the sessile glandular trichomes over a leaf. This is the summary. Cannabinoids are unique compounds found in cannabis plant. They are produced in glandular trichomes. More than 130 cannabinoids were currently identified. The most common cannabinoids are delta-9 tetrahydrocannabinol, called THC, and cannabidiol, called CBD. By the color of the glandular trichomes, it can be identified its maturity. When immature, it is translucent, but later it gradually turns cloudy or yellow and later amber. Immature glandular trichomes are translucent in appearance, while aged glandular trichomes are the peak time of cannabinoid production, appearing cloudy or amber. All right, let's stop here. Thank you. Bye.